Our group is going to present about lipid metabolism. Lipid is a fat-like molecule that does not have the ability to dissolve in water, and it comes in several forms, as shown in the slide. First, lipid absorption. Lipid can be obtained from the daily diet. The digestion of the lipid starts from the duodenum, and the movement of the intestine will break the lipid into smaller droplets. Then, through a series of actions by the bile molecules and the pancreas lipids, the lipid will be broken down into free fatty acids, glycerol and monoglyceride. This fat-soluble molecule will then diffuse into the intestinal epithelial cell and reform into triglyceride. The triglyceride will then pack with the cholesterol and coated with the protein to form chylomicrons and be transported into different parts of the body. Second, the lipid synthesis. The diagram shows schematic overview of pathway involved in lipid biosynthesis in the mammalian cell. From the diagram, we can see that some of the fatty acids must be obtained from a diet, especially the essential fatty acid. Other fatty acids can be produced from acetyl-CoA, which is derived from the glucose. The acetyl-CoA will be converted into palmitic acid by fatty acid synthase, and this is the starting point for other kind of lipid which may have different function in the body. From here, we can also see that the acetyl-CoA can be converted into the cholesterol that is useful for the production of steroid hormone in the body. Third, lipid storage. The lipid will be stored in the adipose tissue. Adipose tissue is the main site of the storage, and they are stored in the form of fatty globules and triglycerides. Cell membranes also store lipids in a small amount. The lipids are in the form of phospholipid bilayers, which give the cell its structure. Lipoproteins serve as a temporary storage for lipids. It helps to carry the lipids in the blood vessel to the different sites of the body. There are 5 different types of lipoproteins in our body. First, the chylomicrons. The chylomicrons function to transport the dietary fats and cholesterol from the intestine to the tissue. Second, the vldl, which is the very low density lipoprotein, the IDL, intermediate density lipoprotein, and the LDL, low density lipoproteins, function to transport endogenous fats and cholesterol from the liver to the tissue. And the last one, HDL, which is the high density lipoproteins, it functions to transport endogenous cholesterol from tissue to liver. The diagram shows the pathway of lipid metabolism. There are 3 pathways of lipids. The first is the exogenous pathway that only involves the chylomicrons. The second pathway is the endogenous pathway, which involves the vldl, the IDL, and the LDL. The third pathway is the reverse cholesterol transport, which involves the HDL. Now we're going to look at the exogenous pathway. The pathway starts from the digestion and absorption of lipid in the intestinal cells. The triglyceride and cholesterol are packed and are coated with proteins to form chylomicrons. The chylomicrons will circulate in the blood vessel, and the chylomicron will interact at the capillaries of the adipose tissue. At this point, the triglyceride will be released into the adipose tissue to be stored. This process is helped by LPR enzyme. The chylomicron will then repackage into other lipoproteins in the liver. Now we are going to look at the endogenous pathway. The endogenous pathway starts from the liver. The triglyceride and cholesterol ester are generated in the liver and packed into vldl particles to be released into the circulation. This is then processed by the LDL in the tissue to release fatty acid and glycerol. The fatty acid will be taken up by the muscle and adipose tissue. One is processed, the vldl will become vldl remnant, and the majority will be taken up by the liver through LDL receptor. The remaining particle will become IDL. Some of the LDL will be reabsorbed by the liver. Others will be hydrolyzed in the liver to form LDL. When the LDL is taken by the receptor, the free cholesterol is released and accumulates in the liver cell. The last pathway is the reverse cholesterol transport. The mature HDL will attract the cholesterol from cell membrane and free cholesterol to the core. The cholesterol ester will then transfer proteins which transform HDL into triglyceride-rich particles. This allows the HDL to interact with the hepatic triglyceride lipids. The rich cholesterol ester-rich HDL may also be taken up directly by the receptor in the liver. The cholesterol ester can also be delivered directly to the liver for uptake without catabolism. The last part of the presentation is the LDL receptor. The LDL receptors are present in the liver cells and are responsible for the binding and subsequently cellular uptake of apolipoprotein B and E containing lipoprotein. The diagram here shows the LDL receptor. The LDL receptor is important to regulate the concentration of cholesterol in the cell. Once the LDL are bind to the LDL receptor, the cholesterol will be released and accumulate in the liver cell. Oversupply of cholesterol will inhibit the synthesis of receptor proteins, which stop the accumulation of cholesterol in the cell. Therefore, the LDL receptor is an important site for the drug to take action to decrease the concentration of cholesterol in the liver. That's all from our group. Thank you.